Hello. Oh, Morgan and Barshan and Susan.
Hello.
Hi, how are you?
Yeah, I'm good.
Good. So, yeah, Barshan, I saw that you had done some, you presented on your materials on Saturday for your recent pull request. Did you
Yeah.
want to go over that again?
No. I think that's all I had for now.
All
I'm
right.
working on something. I'll present it the next week.
Okay. Oh, hi, Susan.
No, been busy around here trying to organize this.
Yeah.
This-
All right.
So I haven't done anything. I'm keep hoping I'll get a chance to one of these days.
So, next week, I talked to Hussein Aether, and next week he's going to present on the work that he um, has been doing. He was collaborating with Dick on the cancer physics work, and he's got some presentation materials on that. So he's going to block off next week, the 10th, and he's going to present during that meeting. So it should be really good. They've been doing a lot of work on that topic, and we'll see what they have. We'll see what the presentation looks like. So it's a lot of medical imaging type stuff, a lot of biophysics. Um, should be good. DevoGraph has been active. We've had a couple of pull requests. I just accepted two yesterday. So this was from Barshan. This is his. This is the interface that he showed in the Saturday meeting. So this is where we have it's Barshan and Claude, and they're putting together. He's putting together this pull request, and yeah. So this was pretty good. Um, and this is for that. And then, of course, we also have Ashu, who submitted her notebooks, and this was for her analysis of the functional data pipeline and lookup table generation. So, congratulations to Barshan and Ashu for that. This was in the DevoLearn DevoGraph repository. So, I wanted to talk about a few papers today. The first one is this paper. This has been getting attention from different people interested in the origins of life. And this was a paper that came out, it was about a month ago, maybe. And it talks about this idea of building a synthetic cell. And they use, in this paper, they use a chemically defined synthetic cell. So what they're doing is they're building sort of this minimal cell, the minimal properties of something that can be called a cell. So we know that one of the tenets of biology is that, cells are sort of the minimal unit of reproduction and of life. So, if we're looking at other than like the very early origins of life, which may have occurred outside of a container, which is a cell, everything occurs in a cell. So even, any kind of single cell organism is usually sort of the simplest forms of life we can find. And so that's what they're working from. But of course, you need to think about, how do you get from, say, like open chemical reactions to closed cellular systems, biochemical systems within cells. And so people have tried to come up with different ways to model that, both computationally, but in this case, they're doing this experimentally with sort of biochemistry experiments. So, people have done things like worked with vesicles. And then now this is the chemically defined synthetic cell. People have also done things with minimal genomes where they've taken existing bacterial cells and stripped out all of the non essential genes, leaving only the things that if you remove them, they would kill the cell. And so they're all different strategies for getting at this. So this is a group here. Kate Adamala, I remember she hosted early life synthetic biology conference. It was at the University of Minnesota, but it was also virtual, and that was last May. I think May of 24 or 25. And I remember attending that, so I was interested to see what this group was up to. And so this is pretty interesting work. Let me go through the abstract, and then I'll talk a little bit about how they define their approach, and give a good treatment of it. So cells are the fundamental unit of life. Yet there is no natural cell for which all its life essential functions are understood. Here we demonstrate a complete cell cycle for a synthetic cell undergoing selection with genome replication, growth, resource acquisition via feeding, and genetically encoded division. So these are, what they're trying to do is build a cell cycle, basically, or something that can replicate. And of course, you need to have a number of things for replication. Some of those things may or may not have existed in the earliest forms of life. So, we think about eukaryotic cell division, prokaryotic cell division, archaeal cell division, and the modern forms that we see in nature probably don't represent sort of the most basal form of this process. But nevertheless, you can try to replicate it and see, what you actually need to have a dividing cell. They demonstrate a complete cell cycle because, of course, is the fundamental unit of life as a cell. You also need to have replication. That's also another fundamental aspect of life. And so the cell is encoded via a 90 kilobase genome that includes functions needed for resource uptake, transcription, translation, growth, genome replication, and division. So they need to have a minimal genome, as I described before, that can do all these things. And of course, you think about if you took those genes away from a cell, of course, it wouldn't be able to do these things. And of course, it probably wouldn't, it wouldn't be able to replicate itself, but it probably wouldn't be able to survive either, because a number of these other things are sort of involved in other processes and metabolic processes. So that's what they're doing. They're getting this minimal genome, and then they're describing this in the paper. The resulting synthetic cell is sufficiently encouraging to support routinization of synthetic cell engineered workflows and will ultimately underlie diverse applications across all biotechnology. So what they're trying to do is they're trying to build this minimal cell, not necessarily to study or to make statements about the earliest life, although you can do that, but as a sort of a vehicle for biotechnology. So the Craig Venter Institute has really spent a lot of time creating different types of bootable cells and minimal cells for this very reason, to build synthetic cell engineered workflows and to be able to just have a workhorse that you can boot up with different things and see, kind of what the additions are able to do. So you're able to build custom functions into these cells. So their first statement here is one of the most ambitious and fascinating goals of bioengineering is to build a biochemical system that could cross the threshold from chemistry to life. So this is where we want to go from the basic reactions of maybe what a cell might do in its initial, in its basic stages, to something that is actually living and can reproduce. And so you need to be able to build these into a cell, or embody them, if you will. Significant progress has been made toward its construction of a minimal living synthetic cell, including approaches based on minimizing natural living organisms, in efforts starting with non-living components. Yet no synthetic cell created from chemically defined non-living components has been shown to implement most life-essential functions, be fully controllable, and described in full operational detail. So this is, again, we need to find what are the most essential functions to a living cell, and can we replicate them? But also be able to control them. So, if we want to control cell cycle in an application, we can do that. And of course, Controlling cell cycle controls growth. So you have a certain amount that the cell grows, and then it divides, and then it keeps dividing, and you can control the size and proliferation of the cell population. These are all things you will use in engineering applications. And of course, we want to be able to describe it as much as possible. Of course, we know from this group that, you know, biology isn't fully describable. I think we've talked about that enough in the group to know that that's probably true. I mean, you have good ways to describe biology, but it's not fully describable in terms of understanding every variation of the process. But that's what they're aiming for here, is an engineering tool. Here we present a synthetic minimal cell with fully defined 90 kilobase pair genome, undergoing multiple generations of cell cycle, selection, and reproduction. So cell cycle, of course, is this process by which the cell goes through different stages of replication. There are different genes that are expressed at each stage. And oftentimes cell cycle is conserved, although sometimes it can evolve in different ways so that different parts of the cell cycle can expand or contract in terms of its timing. Other groups have looked at how to modify cell cycle or how to shut cell cycle off by changing the window of these different phases. So, you know, there are different labeled phases of cell cycle where the cell starts to, you get the formation of spindles, and then you have the division of the membrane, and then you have the final division where you get the two daughter cells. And so you have all these different processes that are interconnected, and you can modify them as you need to in an engineering application. But generally, they're quite conserved in evolution. You also have selection and reproduction, which are, of course, key to different types of population level processes. The resulting gives insights into the minimum components necessary for life. Such a cell might ultimately inspire a chassis that can be adapted for a variety of purposes, framing a foundation for fully artificial organisms. So when they say chassis, you know, they mean basically an embodied form of life, or the cell. And the idea is you can boot up this chassis in different ways, much as you might build like a vehicle of some type. And so this is nice that you can have this sort of minimal form that you can build different things on. And of course, you can create different models or different chassis depending on what you want to do. I don't have a lot of words about embodiment here other than to say I think this is an interesting kind of case study in embodiment, especially with respect to you know, a lot of bio-inspired type robotics and things like that. So just leaving that there for now. Now they say that their work addresses a key challenge in constructing minimal cells exhibiting lifelike properties, and that key challenge is coupling of growth and division to gene expression. So again, you know, like I said, cell cycle is coordinated in terms of gene expression. The different phases of cell cycle coordinate with gene expression. Genes are turned on and shut off as initiators and as terminators of these different phases. So, and the timing is quite, you know, it's quite exquisite in terms of when things get shut off and when things get turned on. So we have these, we have to understand this, and we have to be able to couple growth and division with that machinery. The synthetic cell presented here contains a chemically defined cell-free translation system encapsulated in liposomes. We then combine several previously demonstrated technologies, including genome replication, with new demonstrations of genetically encoded growth and division, selection, and competition for resources. So they talk about how they essentially reconstitute the central dogma of biology. And they say they do this using two major methods. So they use et.al, which is a translation system based on whole cell extracts, and pure, which is protein synthesis using recombinant elements. So I don't have the details on these different things, but I'm not familiar with either one. But they're these protocols that people use for basically creating translation and transcription systems within cells. So this is what they're kind of producing in this system, because it doesn't have, they want to be able to control translation and transcription. So unlike TXTI, pure is a defined reaction mixture with all the components and their concentrations known. So we want to be able to know what those concentrations are. Thus, we used pure, except for early experiments establishing the feeding mechanism. All right, so they talk about DNA replication in liposomes, and extracts have been achieved with the complex bacterial replisome or by a more streamlined method of TT-CDR. And this is transcription, translation, coupled DNA replication. The latter is based on the replicase from the Bacillus subtilis phage, Phe29. We use the Phe29 replicase to carry out rolling circle amplification of the plasmids, constituting the genome of our synthetic cell. And they talk about feeding. So they're supplying or allowing the cell to acquire materials and energy. Liposomes can be grown by fusion using peptide and nucleic acid tags, lipid composition, UV light, or membrane tension. So getting things into the cell, into the liposome, and letting it grow. Synthetic cells can also grow via synthesis of lipids inside the liposome. So you can actually have synthesis within the liposome, coupled with DNA replication, or with lipids from outside the liposome. So you can get things into the cell, you can get reactions within the cell, and so forth. We implement feeding by fusion of nanovesicles directed by peptide tags. The targeting of fusion is directed by a protein encoded and expressed by the synthetic cell. Growth is thus regulated from the genome, coupling metabolic activity to growth and fitness. Selection is another hallmark of life. Artificial selection has been demonstrated in liposomes or in water oil emulsions, which are inorganic. Here we show coupling of the selection trait to synthetic cell growth, enabling selection via differences in the synthetic cell genome. A mutation that results in the production of more protein responsible for growth enables cells with that mutation to grow bigger. More growth results in more daughter cells, thus spreading the mutation throughout the population. Yeah, so this is basically showing that they can do very rudimentary natural selection in these populations. Prior analysis has speculated that a minimal genome for a living cell could be as small as 113 kilobase pairs. The synthetic cells presented here encode a 90 kilobase pair genome. So this is based on prior work in creating minimal genomes. Minimal genomes typically are created by having a genome that is known to be functional. It's usually some wild type from, say, a bacterial genome, and then knocking out genes one at a time to find the things that are only the things that are essential. So knocking out genes one at a time, testing this viability of the cell, and then if that knocking out that gene results in the cell not being able to reproduce or persist, then that is considered an essential gene. And if the cell is able to persist and reproduce, then that is considered a non-essential gene. And then you can build a genome map that shows these genes, and you can create serial knockouts that only contain the essential genes. And so the question is, how small can that minimal genome be? And so the idea is that it could be, people thought it was 113 kilobase pairs. They were able to create something that was 90 kilobase pairs. Okay, so self-replication is another goal of synthetic cell engineering. Several mechanisms for achieving growth and division have been proposed. Recent progress demonstrated reconstitution of protein cytoskeleton, polymer hybrid vesicles, and membrane deformation based on nucleic acids, DNA peptide structures, and other chemistries. Here, utilizing mechanisms based on membrane fission induced by surface protein crowding, we have achieved genetically encoded synthetic cell division in the absence of cytoskeleton. This is interesting because what they're doing is they're initiating some sort of cell division, but they're doing it in a system that doesn't have a cytoskeleton. So they have this, what's basically a protocell, this liposome, or this, it's basically a lipid, and there's no conventional cytoskeleton. So a lot of the things that you would expect the cytoskeleton to do in cell division aren't there. So you need to be able to induce cell division in some other way. And so what they said here is they're able to use surface protein crowding to do this. Taken together, we demonstrated the first minimal cell with a cell cycle, genetically encoded growth and division, all coupled to selection and competition. The synthetic cells undergo gene expression via pure, genome replication via Phe29, and genetically encoded processes of membrane growth, feeding, and division. The introduction of mutation increasing expression of the protein needed for feeding results in faster growth in more daughter cells. This allows for the spread of advantageous mutations throughout the population, demonstrating selection. Okay, so this just gives you an idea of kind of what they're doing. They're building this minimal cell. They're building ways to have these reactions happen. Then they're also trying to induce cell division and some mechanism for selection. Now, given that, we've demonstrated 5 generations of the overall cell cycle, including DNA replication, feeding, and growth. First tested with mechanical division to ensure high efficiency of replication. Utilizing the cell cycle, we introduce an advantageous mutation, increasing expression of the protein responsible for growth. Over several generations, the cells containing the beneficial mutation produce more daughter cells, demonstrating the spread of the advantageous mutation in the population, and thus serves as an example of selection. When cells compete for food in a resource-limited environment, faster-growing cells produce more offspring than the slower growers as food becomes scarcer, illustrating competition for resources driving faster selection. So basically, they're able then to control the reactions, or they're able to account for all the reactions within the cell. They're able then to control the food source, and as a result, they're able to build a system where they can characterize some of these processes in as much detail as they can. Okay, so they're able to illustrate competition for resources. That drives faster selection. Then they were able to engineer a genetically encoded division mechanism, enabling direct link of the genotype of synthetic cells to the number of offspring. And then finally, they were able to couple genetically encoded growth and division, resulting in synthetic cells that can feed, grow, and divide. So they put all those pieces together, and they were able to get a synthetic cell system that mimicked maybe something that you could see in early life, but is really a vehicle for, or also a vehicle for, bioengineering workflows. So a little bit about this. Synthetic cells in this work are liposomes containing DNA genomes encoded across 7 plasmids, an in vivo protein expression system. So these little circular genomes, and then they have this protein expression system that drives the whole thing. Unless otherwise stated, all proteins are expressed inside the synthetic cell. So they have these plasmids, they have this protein expression system within the cell, and then everything is expressed within the synthetic cell. The synthetic cells presented herein have a defined chemical composition at the time of formation, with known concentration of components making up the cell, starting from the pure system. And of course, their synthetic cells divide without cytoskeleton. Division is first achieved by mechanical extrusion through a membrane filter. Ultimately, we demonstrate genetically encoded division as a result of membrane protein interactions. So this is like they kind of go over this in the figures, but basically they're inducing division, and then there's this protein crowding on the membrane of the liposome, which is not a cytoskeleton, but is a lipid layer. Okay, so this is figure 1. This is a cell cycle of synthetic cells with a 90 kilobase pair genome undergoing selection and replication. So this is what it looks like in the cartoon. So we have some green fluorescent protein images here, where we go from 1 over to 6, and you can see that it's hard to see. I'll zoom in. The green is really hard to see against the black background, but basically you have a single cell here, and then it starts to divide by things going to the poles. You get the spindle formation, and then the membrane starts to pinch, and then you get division. Notably, it doesn't have a cytoskeleton, but as far as I know, there also aren't complexes within this that are pinching off the cell, the two daughter cells. So I'm not really sure exactly how that whole process works, other than through some sort of membrane crowding. So they're kind of coming up with this mechanism for doing this. But anyways, at the end, you end up with 2 cells. And so this is what your cell, your liposome looks like. So you have, this is one of our plasmids here where the genome is encoded. It's a circular genome. And it's transcribed here with an RNAP. And then you get this mRNA. These free-floating mRNAs then go through this protein synthesis method where you get protein synthesis. So you have this 90 kilobase pair genome. You have genome replication, gene expression, and then you have the interaction with this membrane channel. And these membrane channels then go to these are proteins that go to the surface of the liposome. They do things like waste removal, nutrient uptake. And then you have a generation counter where you have this RNA ligase that ligates different generations of RNAs. And then you have selection on these cells. You have slower growth and faster growth mechanisms. And so that's basically the process by which you have the cell operates, and then you have cell division. So there are a lot of notes on this figure. Figure 2 is genetically encoded feeding and growth of synthetic cells. So here you have a couple of populations here. So you have 2 populations that are fusing together. And so you have this fusion event where you have population 1, which is a T7 RNA polymerase, and then population 2 with the GFP plasmid in it. And so now you're combining them through fusion, and you have these membrane proteins here that are joining them together. This shows membrane mixing, so you have population 1 versus population 2. This just shows the fluorescent signals, GFP and cytochrome 5, so you have these different intensities here. Actually, the ratios of the plasmid and tag. And this is just as a verification mechanism to make sure that this process is happening the way it should. Then you can also look at different ratios of cells. So this just shows that. This is different ratio of cells using a GFP signal, GFP mRNA, DNA, and fluorescence, and just showing this. And then these are the microscopy images here of the different cells in the population. So you have the red signal, which is this signal here is cytochrome 5, and then the green, which is the GFP. So you have the two channels separated and then the two channels overlapped. So you can see that they're like that. And this just shows microscopy images of liposomes after fusion. The green channel shows GFP expression. The red channel shows rhodomine, membrane dye. And the third panel in each grouping is composite of the red and green channel. The liposomes are from experiments under the same fusion conditions as data on panel D, with images collected after 12 hours of incubation. Okay. And in this case, they're using protein expression data using the whole cell lysate et.al. Figure 3 are 5 generations of cell cycle with 90 kilobase per genome. So this just shows the cell cycle over this 5-generation window. This just shows, I guess they're sampling different generations here. So they have 3, 4, and 5. And they have new DNA and mRNA. So they have DNA synthesis and then mRNA. And then they have translation into proteins. So they have, it's a pretty busy figure. This is Phe29, which is fusion. No Phe29 also with fusion. Phe25, no fusion. And no Phe25, no fusion. So you can see in these 4 conditions here what you have for these different slices here. You have GFP, alpha HL, PLD1, PLD2, PLD3, PREP, and T7 RNAP. So all these components within the cell, and then they show that you have this in these different conditions. So all of these are shown in Phe29 fusion and Phe29 no fusion. So this is the first Phe29 fusion is DNA replication, feeding, and growth. And then Phe29 no fusion is DNA replication, no feeding, and growth. So you see this pretty consistently across these different assays. And then you have mRNA, which is GFP, alpha HL. And then you have your generation counter, which is for each generation. So this process, you have for this here, for the measure here, in Gen 3 you have Phe29 fusion and no Phe29 fusion. And then in Gen 4 and Gen 5 you don't see much of that. And then for translation, which is the production of proteins, you get Phe29 fusion, which has a higher RFU signal than no Phe29 fusion, and then in descending order, Phe29 no fusion, and no Phe29 no fusion. So you get this step down. I don't really know how to interpret these other than to say that these are the graphs. You see similar results in generation 4 and in generation 5. This generation counter, I'm not sure why this is across these different, like you have 3, 4, and 5 across the generations, but in any case, those are the data. And we can dive more into that if you'd like, maybe offline. This is selection in populations of synthetic cells. So this is where you're getting this selection. You have this cell fusion here where you have the T7 RNAP plus the plasmid, and you get this GFP that's expressed after fusion. And so this just shows some of the details of that. And then here you have 5 generations of competitive growth and division, which we just saw on the last slide. This is where you have your cells that are fusing, and then they're dividing, and you have this process here. Figure 4 is selection in populations of synthetic cells. Or I'm sorry, I think that's what we just looked at. And then figure 5 is competition for resources over multiple generations. So this is where we have competition for feeding resources, we have cell division, and we have our feeder liposomes, which are fused to the cells, and then we have this competitive process. So this just shows some of the results here. And then figure 6 is genetically encoded replication of synthetic cells. So this is genetically encoded division. This is where we have our membrane proteins, and they crowd the membrane so that you have division events. And then you have this strep-dividin binding membrane protein where you're able to use this to induce membrane curvature and get division. So you have these membrane proteins that crowd the surface. You have these strep-dividin molecules that kind of pull it apart, induce curvature, and then you have division. So it's kind of a nice way to do this. And they're actually using click chemistry in this process to get this to work. And then they just show genetically encoded growth and division. So you have these division events here illustrated. And that's just showing that sort of the efficacy of this process. And that is it. Those are our figures. So the 6 figures that show that process. So a nice paper going over some tools for early life and for bioengineering workflows. Okay. Do we have any questions or comments about that paper? So the second paper I'd like to talk about today is this paper, A Blastoporal Organizer and a Ctenophore. So this is a paper, it sounds like I have to explain the title, but all it really is talking about these conceptual things in developmental biology. One of them is the organizer, and the other is the inducer. So in the history of developmental biology, in embryogenesis or in embryology, people were trying to figure out how embryos formed patterns. And last week we talked about a paper where they did some experiments and they did some modeling with different pattern formation models, mathematical models. And here they're going to reconsider some older work. And so they're looking at the role of the organizer. They're looking at what the organizer does, and then they're replicating this in another type of system. So let's read the abstract and see what they're talking about here. So in an iconic experiment in 1924, Hilda Mangold and Hans Spemann established that the dorsal blastopore lip in amphibian embryos function as an organizer and induce a secondary body axis when transplanted into a host embryo. So an organizer is one of the things that is a chemical signal that does this job of organizing things within the embryo. So, we talk about self-organization, and we talk about how it's usually the product of a bunch of things interacting, and those interactions produce some sort of emergence. It could be weak emergence or strong emergence. Weak emergence is kind of being patterns that emerge from unspecified interactions, and strong emergence being something maybe driven by a process. So this might be an example of strong emergence, but they didn't really talk about it that way. First of all, they didn't have the language, but also because they were thinking about this in terms of the chemical signals in the embryo. And so the thing that they proposed was this organizer. And so this is where, you have this agent that is basically organizing order in the embryo, starting with the axis, or one of the anatomical axes. This discovery demonstrated that specific embryonic regions can regulate embryonic patterning and lead to the establishment of an entire body axis. So this is where we have this body axis that we want to establish. And so what they did in the experiments is they actually induced a second body axis. So we have usually we have an anterior posterior axis, and then we have a left-right axis, and we have a dorsal ventral axis. We have usually 3 axes, but usually those are very distinct. And so this is where they induced an additional body axis in the host embryo. But the idea was that if you induce this signal, you can basically induce this kind of pattern. Okay, so subsequent studies have revealed that cnidarians, the sister group to bioRxiv, so we're bioRxiv, we have a left side and a right side, and those are symmetrical for the most part. And our sister clade, or our sister taxonomic group, is a group of organisms called cnidarians. And of course, because they're not bioRxiv, they don't have this left-right axis. So, and they have a different organizational scheme. And so the original experiments were done in bioRxians, and so you were able to induce an additional axis there. In this case, we're looking at cnidarians, and we would like to also see if they have a blastoporal embryonic organizer, and indeed they do. This came in subsequent studies. However, the evolutionary origins of the organizer cnidarians remains unclear. Here we report that the blastopore lip of the ctenophore, Nemeopsis ladei, or lady, Nemonopsis lady, a member of the evolutionary sister group to all other metazoans, exhibit organizer activity. We show that transplanted fragments of blastopore lip tissue from M. lady gastrula introduce secondary pharynx and mouth formation. So again, we have the same result in the ctenophore, where we have, which is a cnidarian, which is where we have the secondary pharynx and mouth formation, which is, of course, organizing a secondary process in the embryo. Moreover, transphyletic transplantation experiments show that the blastopore lip of M. lady leads to the generation of a secondary body axis in embryos of the cnidarian and Nematostella vectensis. Organizer function M. lady requires both beta-catenin and TGF-beta signaling, and the TGF-beta family ligands probably provide this inductive capacity. So this is where our organizer is really a group of chemical signals that basically act in a cascade, and we know that this whole family of TGF-beta are involved in this process. These findings reveal the deep homology of the blastoporal organizer in ctenophores, cnidarians, and vertebrates, implying the ancestral organizer of the blastopore lip. We propose that the emergence of the organizer was an essential innovation that facilitated the change in the temporal cell differentiation of unicellular relatives to the spatial cell differentiation of the first multicellular embryo. So this is about spatial cell differentiation and about how we go from a bunch of cells in a cluster to these patterns. And what this study shows, or what this paper is discussing, is the idea that you see this in bioRxians, but you also see this in other sister clades to bioRxians. And this has a common evolutionary origin, which is in this TGF-beta signaling pathway, which is highly conserved across those taxa, across those lineages, and they have this inductive capacity, which is, they say, probably provide. So we have sort of a candidate for our organizer, for what we might call our basal organizer. So okay, that's the abstract. And they talk a little bit about the details here. So the concepts of the embryonic organizer and induction were introduced and developed by Mangold and Spemann based on their landmark experiments in the 1920s. Their studies on transplantation of the dorsal blastopore lip of amphibian embryos demonstrated that this tissue had the capacity to induce the formation of a secondary body axis by recruiting neighboring host cells. So this is a process where you have this body axis that's already established or is being established. You add in this inducer, and it's able to take host cells, pull them into this new process, and do more, have a secondary organizing function. So the secondary axis is formed through interactions between the transplanted cells in the experiment and the host embryo tissues, and the data highlighted the critical role of the organizer in directing host tissue differentiation and morphogenesis. This experiment provided clear evidence that specific cell populations in embryos can influence the differentiation of neighboring cells.
Should I say what Dick said about the organizer?
What did Dick say about the organizer?
He said that they put all kinds of things in the blastocele to see what would organize, and they put fly wings in, that I remember that, and a bunch of other things. And the things that acted like the organizer, he noticed, were polar. So they had a charge. So he says that was what it was, is if you had a polar molecule and you put it in the blastocele, it would act like an organizer.
Oh, wow.
Yeah, it's just an observation he made, and he told me about it. So, yeah, so perhaps the reason why the organizers work is because a very good polar molecule is created by some of the cells. And I do know that if you have the wrong size or shape of a blastocele, it will ruin the development of the long axis of the animal. At least I think that's what's going on with it. I haven't got a chance to do this experiment, but the axolotls, there's a morpho axolotl, and when you have made a morpho with a morpho, you get eggs that don't develop properly and usually have bent tails and die. And we looked, the lab that I was associated with did an optical coherence tomography look at the inside of the egg, and the blastocele was small and right near the edge. So it was the part that develops into the animal was smaller.
Okay.
So I think it probably has something to do with this. I would like to work with it, but trying to find someone who's interested in this is a bit of a stretch. It's persuaded Dr. Sharif that he could get a student to take just ordinary axolotl eggs and get the image of the interior of them. So I hope he can find someone who's interested.
Yeah.
So anyway, that's a long explanation about that, but
yeah.
Yeah, so oh well.
It's okay. Yeah, it's good. No, that's good. Actually, it gives a lot of context to what they're talking about here. Basically, this experiment, of course, provides clear evidence that specific cell populations in embryos can influence the differentiation of neighboring cells, which is something that was replicated again and again throughout the history of developmental biology, where you can do experiments where you can take cells and you can implant them in a certain cellular tissue context, and you can get them to change to that cell fate. So usually we're talking about some sort of precursor cell or a stem cell, and you implant them, say, in muscle cell, and those cells will become other muscle cell. Sometimes, of course, you get, different, and of course, this has to be in an embryo that is not, that is a regulative embryo or a regulative process governing this. Um, and so, this is just showing that there's an influence of these signaling molecules on the cells. And so the same thing is happening here where you have this organizer, and they say in the paper, they make the statement that it's some sort of cell extract or aggregate, which is just a fancy way of saying we're just getting some cells from somewhere and putting them in there. It's kind of not as glamorous when you put it that way, but that's basically what they're doing. And, this is just basically a non-native signal in the embryo. And so you're able to induce this new axis given that it's the proper signal. So whatever the signal is, that's usually where the system goes. And so that's what they're talking about here. Subsequent comparative evolutionary studies revealed the presence of an embryonic organizer in a non-bilaterian animal, or the cnidarian, which we talked about, transplanting a section of the blastoporal lip from an early gastrula of N. vectensis to another embryo at the same stage of development results in the induction of a secondary oral aboral access. The central role of the Wnt beta-catenin signaling and driving the organizing activity of cnidarian and vertebrate blastoporal lips suggests the potential homology or the shared ancestry of these structures. Despite extensive research on the embryonic organizer in bilaterians, particularly chordates, and in this case they're talking about the zebrafish and frog Xanthopus laevis. So those are, of course, well-known experimental model organisms. And then their sister group in the cnidarians, which is N. vectensis. The evolutionary history of the embryonic organizer remains unsolved. So they're actually looking at this evolutionary, sort of shared evolutionary ancestry of organizer activity in both like cnidarians and bilaterians. And so it is still unclear whether embryonic organizers exist in other non-bilaterian animals, predating the cnidarian bilaterian separation. So those include organisms like sponges and comb jellies. And so comb jellies are an example of ctenophore. Ctenophores are of particular interest because they are considered to occupy a key phylogenetic position as a sister group, which means they're sort of in a different phylogenetic group, but they're related, the most closely related to all other multicellular animals. A ctenophore embryonic organizer, if homologous to those in cnidaria and bilaterians, would suggest that an organizer emerged alongside the advent of multicellularity. So that's really what they're getting at is the organizer sort of a hallmark of all multicellularity that sort of evolved at the same time as multicellularity? Or is this just something that has different mechanisms and is does not have a shared ancestry? Okay, so this is figure 1. This just shows embryonic organizers in different metazoan groups. So this just shows you a bilateria up here, represented by a frog, cnidaria here, represented by a comb jelly, and then you have these earlier groups, Porifera, ctenophora. These are outgroups to the cnidaria bilateria group, and it just means that they evolved. They separated from the common ancestor or the common ancestral lineage earlier, and then Coenoflagella or Coenoflagellata, which is out here as an outgroup. And so these blue lines show that there's this putative embryonic organizer. So in cnidaria and bilateria, we know that there's an embryonic organizer present, and they're showing this common ancestry here. So the common ancestor of cnidaria and bilateria, and then the common ancestor of Porifera and cnidaria bilateria is back here, but we don't know if the embryonic organizer existed there because there's no evidence of an organizer in Porifera, nor is the case of ctenophora, where there's no evidence of the organizer. So we suspect that the common ancestry of the organizer is at the cnidaria bilateria split. So this just shows sort of what the organizer is doing here. So in this experiment you have, so this is an example of this experiment where you have an embryo where you open it up and you put in this cell extract, and then you have, as the embryo develops, you get these two axes. So you get this primary axis and a secondary axis, as you can see. You put the extract into the inner cell mass, and you end up with the secondary axis. Kind of interesting kind of experiments that you can do in developmental biology. So this is in the bilateria, where you have this secondary axis form as a result of putting this. So you're just clipping out a piece of this cell from the embryo here. You're putting it into the inner cell mass in this other embryo here, and then you get these two different axes form. In the cnidaria, though, of course, it's a different anatomy, but you're still able to cut a piece out of this embryo and put it into this embryo here. And that means that you get, because, of course, cnidaria has a vastly different type of anatomy, you get this primary axis here and then a secondary axis branching off here. So you get the same effect. And the idea is that the organizer is acting locally here versus globally here, and it's kind of organizing its own set of fates. So the example I gave before of putting like stem cells in a muscle cell and having them sort of acquire that muscle fate, if you transplant the right materials, if you induce an organizer around those materials, you can get sort of secondary organization. And that's kind of what they're showing here. All right, so this is just an example of what they did. And these are these classic experiments, these transplantation experiments. But what they're saying is that these two things are connected by the same set of organizers. And so an essential characteristic of an organizer is to recruit surrounding host tissues to form induced structures after transplantation. To explore this effect in our experimental model, we transplanted pieces of the blastoporal lip, which were labeled with the vital membrane dye. And they talk about how they labeled these in host gastrula embryos. In transplantations that resulted in the induction of a fully developed ectopic pharynx, the pharyngeal tissue is composed of a mix of both host cells and transplanted cells. However, the distribution of donor and host cells in the induced pharynx varied. In one instance, the ectopic pharynx consisted of donor and host tissues with a sharp boundary between them. Alternatively, only one wall of the ectopic pharynx contained host-derived cells, or the induced pharynx was composed of a uniformly mixed population of donor and host cells. So they show how the host cells and the donor cells balanced out across the anatomy of the developing embryo. So notably, host cells were never observed in the area of the ectopic mouth opening. This kind of split between donor and host contributions is also seen in classical mangold Spearman grafts, which is sort of the classical experiment. And sort of if you replicate that experiment, you see this kind of split between the donor and host contributions later on in embryogenesis. So donor cells largely contribute to the axial midline. So the ectopic notochord, which is almost entirely donor-derived, versus surrounding parts of the induced axis, which contain a mixture of donor and host cells. So depending on, you basically put these into the same embryo, you put a chunk sort of in one part of the embryo, and the rest of the embryo is the host, and then that chunk that you put in is the donor. And then as the embryo develops, of course, then they start mixing in certain ways, but also maintain sort of their own identities. That's kind of interesting, but not surprising given what we know about cell migration and shape transformations in embryogenesis. Okay, so that just talks a little bit about what happens when you put these in these transplantation experiments and sort of the downstream effects. Here we determine that the phenotype described is a mouth-like structure without a developed pharynx, and that results from a failed induction as the structure is composed exclusively of donor tissue. So there are phenotypes that are inviable and that result from failed induction. For transplantations in which the blastoporal lip graft did not induce an ectopic pharynx, we selectively labeled donor cells to trace the distribution of the transplanted tissues. In these embryos, we observed considerable variability in the distribution of donor-derived tissue. Donor cells were incorporated entirely in the primary pharynx, scattered across the superficial epithelium, or integrated into the tentacle bulb and tentacles. Thus, the formation of the secondary pharynx and mouth following blastoporal lip transplantation in ctenophores involved the recruitment of host cells in the ectopically developing structure, which is the hallmark of a bona fide organizer. So basically, the organizer should allow for the recruitment of host cells in different ways, and we expect to see that type of activity in an organizer. So there's a specific type of behavior we expect out of an organizer to say something is actually an organizer. And so then they kind of talk a little bit about the molecular biology of this process in ctenophores, where they saw expression of different factors. And this is just a map of that where they're showing this graft, and then they're showing this organizer activity through the expression of these different factors. And this is actually a nice figure showing kind of the ratio of embryos where they get a single pharynx, two pharynxes, and then a secondary incomplete pharynx. This is figure 3, showing that in ctenophores, the embryonic organizer can induce secondary axis formation in an idarian embryo. So it's not always the case that you get the secondary axis formation, but in this shows you how this works when it's successful. So again, you have a donor. In this case, it's M-lady, which is this ctenophore, and you're giving this piece of the developing embryo to the cnidaria envectensis. This is a blastoporal lip graft, and you're putting it inside here. You also have this lateral ectoderm graft, and it's just showing the effects of this. So it's across orders, and you're actually able to induce this secondary axis formation. Okay, so this is just showing a little bit about the signaling, and so it involves this TGF-beta pathway. They talk about TGF-beta SMAD signaling, and activated by nodal and activin ligands. And this is actually where there are small differences between ctenophores and bilaterians in terms of the signaling pathways, which of course again is expected, but they do share these deep evolutionary roots that may exist in pre-bilaterian lineages, but how far back that common ancestor goes, we don't really know. We know at least it includes ctenophores with this paper. And then this just shows that in, like you might ask, if we have our TGF-beta family ligands, if we express them in the blastoporal region, if we overexpress them, is that sufficient to induce pharyngeal branching and mouth duplication? And this just shows the sort of what's going on here. So in this this example A, we have the ctenophore M-lady. You have this gene cloning experiment where they clone the gene, they have this mRNA injection, and then you have this this embryo that then they show they basically overexpress this, and then they inject the mRNA into a cnidarian cell. So this is actually where they have this, they're taking out this extract, they're cloning the gene, and they're injecting it into one of these cnidarian cells. And so from that, they're able to build this model. This is a proposed scenario for the emergence of the embryonic organizer and the common ancestor of all metazoans. So this just shows your phylogeny here, where you have some putative unicellular ancestor. And so these are all the multicellular orders of metazoa here. You have ctenophores, porifera, cnidarians, and bioRxiv. And then they propose here is the origin of the blastopore associated embryonic organizer. You see this in ctenophores and cnidaria. So what you have here is you have cnidaria, where you're demonstrating this, but you also have ctenophores, where you're getting your source materials for these ctenophore cnidarian experiments. We know about bioRxiv, we know what's going on there. And then porifera, we don't have any data, but we suspect because ctenophores are involved in this sort of these organizer experiments that the origin of the blastopore associated embryonic organizer lies at the common ancestor of all four of these groups. So this is the support for the statement where they say that the sort of the common ancestry of this organizer lies sort of right at the evolution of multicellularity. Okay, I think that's enough for that paper. I think we get the idea. Okay, so any questions or comments about that paper? I know it was quite a complex paper, but Susan?
Oh, it's just, I think Dick's comment about polarized molecules causing some of this is true. It's definitely an observation about it. And I think he was looking at some paper that tried various substances into a blastocele to create like two heads.
Yeah.
Yeah. And the other person who's done work on blastoceles and embryos in general is Lev Belousov.
Oh, right, yeah.
He's a Russian, so that's another paper that would go with this study.
Yeah.
And I hope to do some work on this. I need to find someone in the biology department maybe who's interested. I don't even know how to go about that.
Oh. Yeah, you mean getting in touch with them or doing the work?
Yeah, getting in touch with them, talking to them. Yeah, no, I'm in engineering, and it's well, I could talk to the biosystems engineers, I guess. They're working with the guys who do insects, the entomologists. Anyways.
All right, I think that's all for
The
today.
blastoceles are interesting.
Yeah.
And those are all internal, what I call internal blastoceles. I don't know. There's different types, just so everyone knows. There's the animals in amphibians, they develop on the surface with the blastocele interior to them. And in humans and mammals, they develop inside the blastocele. The blastocele covers them.
Yeah.
They're different types of blastoceles. Different configurations.
Right.
That make different animals.
Yeah.
Of course, and then there's fish, but Yeah, I'm going to have to leave early next week, so I hope I'll get to see his presentation first thing.
Yeah.
I
Hopefully.
might be on the. I'll be on the East Coast, so we'll see. But
Okay.
I'll
You'll
try
record
to.
it,
I'd be
right?
very interested
Oh
to.
yeah, yeah.
Yeah.
All right. All right. Thank you.